Hello everybody, this is the lecture for chapter 8. We're going to be talking about reproduction, cellular reproduction, and then we'll talk about a little bit mating systems later when we come back for in-person. So, reproduction. What do we mean by that? What do we mean by that is looking at creating a new cell or an organism capable of life on its own. Now, for a single cell organism, this is like a yeast here, reproduction and cell division are essentially the same thing, right? It's just the creation of a duplicate cell, but what they're going to be doing is they're going to be dividing and copying everything in themselves to make a whole new cell, which then essentially makes a whole new organism. Now for larger multicellular organisms, that becomes a little bit more complicated, and we're kind of going to go through that here. So, the most simple form of reproduction is asexual reproduction. This would be reproduction without fertilization, where all the DNA, or in this case what we're talking about, chromosomes, right? Remember, DNA are packaged in these things called chromosomes. All the DNA comes from a single parent. So this little amoeba here we can see dividing into eventually two amoebas. A strawberry plant has these little runners that come out, and they, go a couple inches away from the plant, and then a little new baby plant gets nutrients sent to it through that little runner, and eventually roots grow from here, and once this plant gets a little bit established, then this runner here will disappear. But, animals can have asexual reproduction too. This is a aphid, where a little baby is coming out, and this is a female, and this is an exact genetic copy of her. Aphids are just always asexually reproducing. This is a sea anemone, where we get a little baby budding off from the main mother plant there. So we're going to go through this thing called mitosis and meiosis. Mitosis and meiosis, sorry. Now, in asexual reproduction, all cellular division is done via mitosis. And what that means then, what that ends up with, is the parent and the offspring have identical genes. Now, sure, there might be some mutations or some chromosomal defects or something, which essentially is just a mutation. But, barring that, all the DNA is going to be about the same. Thing is, when you see identical twins, right, true identical twins that are genetic copies of each other's, they can be slightly different, right? They look different. Maybe if one has an outside job, another has an inside job, the outside worker might have a little darker skin, if they tan well or something like that. So, environmental effects can differentiate organisms even with identical DNA. So let's switch over then to think about sexual reproduction. Sexual reproduction is the fusing of two gametes. So for humans, we know this to be sperm and eggs, right? With plants, essentially pollen. This is a super close-end picture of what pollen is. And this is a certain type of pollen. I'm not sure what species it is. Doesn't really matter. But inside that pollen is actually the sperm cells. Now, we often think about the world as male and female. But there's other ways to do that too. What we see is fungi. Fungi have mating types, which doesn't really correlate to male and female. But what we oftentimes see is that fungi will have many different sexes that might have 20, 30 sexes, and as long as they meet up with someone or another fungus of a different sex, they can mate with them. Now, sexual reproduction requires a special type of cell division here called meiosis. Think about a human. An adult human has 46 chromosomes, 23 from mom, 23 from dad. But you can't just take all of those chromosomes and give it to the sperm and the egg, because then, if you have two parents with 46, if they were to give 46 and 46, the baby would have 92 chromosomes. That'd be a problem. So meiosis, what it does is it halves the chromosomes. So this sperm here has 23 chromosomes. This egg has 23 chromosomes. When they combine, they'll create a baby with 46 chromosomes. And that going from 46 to 23 is this thing called meiosis. So, which is better? Asexual reproduction or sexual reproduction? Now, usually when I ask this question, most classes, most of the hands go up when they vote for sexual reproduction because, that's essentially what humans are, right? But there's actually some scenarios where asexual reproduction is nice. Why would you want to do sexual reproduction? It's expensive, right? You have to buy your fancy clothes and have a nice car and go on a date and pay for dinner. That takes a lot of time. You can expose yourself to danger. These two male deer are fighting, and their horns or their antlers got locked up, and this one died. This one probably will die relatively soon if it's not helped by whoever took this picture. Having this ridiculous inflatable sack here on this frigate bird, that's expensive, and it's just kind of a waste of time and energy and resources to make this thing, except there are some good things that we'll talk about sexual reproduction. But I think these things show that it's expensive to go out on dates. It takes time and it's dangerous. And if you have an individual that is perfectly adapted to an environment, it might be nice to make an exact copy of them, right? How would you get that advantageous combination of genes again in the future? You might not be able to. So recombining to have your chromosomes down from in humans from 46 to 23, that can be destructive. You're essentially giving up half of your DNA for your offspring. So why would you want to do asexual reproduction? I should say you want to, right? We don't have a choice. Animals don't have a choice for this. But maybe we should ask, where is asexual reproduction advantageous? And it's quick. It's inexpensive. It's not dangerous. You can do it in the safety of your own home, right? The bad things are, this Daphnia here, it's this little water flea. It's about less than a millimeter long. All these little eggs here that will turn into little Daphnias will be exact copies of her. And what happens if she has a mutation? Well, all of her babies have a mutation. And that mutation stays in her line forever, right? So there's this idea of Miller's ratchet. A ratchet is a tool that, you can only tighten. And, think about a crescent wrench that as you tighten and then you turn it back, it goes click, 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 right? And you can only tighten and make the bolt tighter and tighter. And that's what's happening. Every time there's a mutation in an asexually reproducing species, that mutation ratchets it closer to extinction. Think about spreading then a beneficial mutation. Well, you can only spread it to your offspring, and it's hard potentially to get those, get that good mutation out. Then, if everybody's the same, right? Let's say, she has these babies. Let's say she has 100 babies in her life, right? This little Daphnia then, if one of her babies is susceptible to a disease, that means every single one of her siblings is susceptible to a disease, right? If everybody is the same, if one person gets sick, then everybody else will get sick, if they're all genetically identical, right? So when we see this sexual versus asexual kind of thing, essentially there's pros and cons to each one. And some organisms actually use both. So what happens with Daphnia, these little water fleas, is from the spring through the fall, basically, they reproduce asexually. There's just females that will reproduce asexually. But then when winter comes around and their food starts to be limiting, what they do is they switch over to sexual reproduction. They'll produce some of the females will produce eggs that will become males, and then when those males mate with the females, they produce resting eggs that will then hatch out in the spring. So not all organisms are just stuck with one mode of reproduction. The thing is, the benefits of sexual reproduction are very, very good. When you have two sets of DNA, right? Think of your DNA. Half of it comes from mom, half of it comes from dad. If dad gives you a bad mutation, oftentimes the DNA from mom can mask that bad mutation, what we call a recessive allele, and you will appear normal. You'll have the normal trait or something. DNA, when they line up next to each other, they can actually repair themselves. Like this homologous chromosome idea is when, let's say you have a chromosome from mom for eye color and there's a problem with it. But if that chromosome from dad, which also has eye color on it, line up next to each other, essentially your cells sometimes can like proofread it and be use the good chromosome to like fix the broken one. But by far the most important thing is this generates diversity, right? Even look within your siblings, if you have siblings, right? Or look at sibling pairs. No individual sibling is, or no two siblings, even if they have the same mom and dad, right, are going to look the same. Now that's true for humans, that's true for dogs, that's true for any organism you want to think about. And this diversity is really, really good because it allows, like, basically that old saying, put your eggs in more than one basket, in more than one genetic basket. So everybody's a little bit different, and that's a good thing. So what we see is that sex dominates in the vertebrates and less so in other groups, right? So we see a lot more asexual reproduction in like worms and snails and things that don't have a backbone, insects. Now, let's get into this idea then of thinking about how cells are dividing. So before we do that, we need to think about some terminology. So diploid. What diploid means is that a cell contains 2 copies of each chromosome. So that's what that's referring to is essentially 23 chromosomes from mom, 23 from dad. They're in these things called homologous pairs, meaning the chromosome 1 that you get from dad might have eye color, hair color, and I don't know, some other trait regarding how curly your hair is. Okay. Now, did I say from dad? And then from mom, that chromosome would have the same things. It would have a trait for hair color, hair curliness, and eye color. So they have the same information on them, but you just have 2 copies from that. We often like abbreviate that as 2N. Now, haploid, a haploid cell is a cell that contains 1 copy of each chromosome. And that's really what we're talking about here are the sex cells. Things like sperm and eggs. Okay? Now, you only find haploid cells, at least for humans, in the sex cells, in the sperm and the eggs. Now, when a sperm and an egg meet, then, we get this thing called fertilization, where the egg, a sperm comes to the egg, and the egg lets that sperm in, essentially, and the DNA gets fused, the chromosomes come together, so it was a haploid egg and a haploid sperm that come to create a diploid zygote. A zygote is a single cell that has been fertilized. Now, then there's 2 processes by which cells can divide. After fertilization, and we create a zygote, a baby will undergo mitosis. And mitosis is basically all of the growth in an organism. And we won't get meiosis until that baby grows up and produces either sperm or eggs. That's the cell division that results in haploid sex cells. Okay, so what we see then is how a cell grows is that basically most of its life is spent in this thing called interphase. Now, interphase is basically what the cell is doing most of the time. Okay? A skin cell is being a skin cell, is protecting the skin. And this is the time of what we would consider like normal cell function. What's happening is that in this, the DNA needs to, well, basically the cell needs to grow, the DNA needs to get copied, and then everything else needs to get copied. So it needs to have, basically double the number of mitochondria, double the number of endoplasmic reticulums, double the number of ribosomes. If a cell is going to divide into 2 new cells, it needs to, essentially be twice as big. It needs double the amount of DNA. Then it goes through this thing called a mitotic phase, which is the nucleus dividing, basically the DNA being pulled apart into 2 copies. And then the cell needs to divide in this thing called cytokinesis. So, if you look at a cell, what you see is mitosis. When it's going through this mitotic phase, it's really accurate process. There's only errors in 1 out of 100,000 times that this happens. Now, mitosis is unique to eukaryotes because what we're going to be talking about is chromosomes. And remember, only eukaryotic cells have chromosomes. So this is really like a continuum of things, but we divide it into 4 phases. The mitosis into prophase, metaphase, anaphase, and telophase. So, let's think about, though, before mitosis, which is this thing called interphase, where the cell is growing, everything is doubling. It's doubling the materials, doubling the organelles, doubling the size of the cell, and doubling the DNA. At this time, remember, chromosomes are loosely packed as that thing called chromatin that we talked about the other day, where we think about DNA as being as like an open book with all of the pages laid out on the floor. That's analogized here by these, diffuse lines. This is like the DNA that is like all the pages laid out on the floor. It's not all wound up and coiled into a chromosome. When we get into prophase, then, what we see is the chromatin condenses into these things called chromosomes. And that's what we generally think of. Here's chromatin. Here, the blue lines here, then, are the chromosomes, which are these little X-shaped things, or at least the things that we kind of draw as X's. And that is that is where all the DNA has been like closed up. All the pages of DNA have been closed up in the book. The chromatin has condensed down and wrapped up on itself. Now the nuclear membrane then, that membrane that houses the DNA, has to disappear, right? If we're going to cut the DNA essentially in 2, we need to get rid of that membrane. What we see then is this DNA, when we draw it as this like this X kind of thing, or maybe like 2 lines kind of next to each other, that's called a sister chromatid because the exact same information is on this side that is on the other side. Then in metaphase, those chromosomes line up in the middle of the cell. What we should remember, it's not just a line, it's a 3-dimensional structure, like, there's a bunch in here. And these microtubules that are part of the cytoskeleton then attach to those things and continuously like pull on each other to keep these chromosomes in the middle. Then in anaphase, those chromosomes separate and become what we call daughter chromosomes, right? So this now has all the information that is on here. These are completely identical. These microtubules will pull, these microtubules will push, and it kind of makes the cell kind of like football shaped. And by telophase, then the chromosomes reach the separate sides, and then the nuclear membrane kind of folds back, or appears back and is built around the DNA to create a new nucleus. Then finally cytokinesis happens, which is the division of the cytoplasm. Now this essentially starts before telophase is totally finished. I should say, I have 2 videos here. I skipped over one earlier. I'll put those in the description of the video, so please take a look at those so you can get, like a whole kind of explanation of how this goes on. Now you might be asking, well, how does cytokinesis happen in plants, right? When you have a cell wall, sure, the cell can get bigger, but it can't like pull apart from each other. The cells can't really elongate that much. What we oftentimes see is think of this as like a room, okay? Now, if you want to make this into 2 rooms, right, to make a new cell, what you could do is essentially bring bricks into the room and just start piling up bricks and mortaring them together in the middle of the room, and to make like basically a cell wall here, which becomes this like plate, which eventually you can divide the cell into like 2 cells, and you get a new cell that way. Okay, that is mitosis. Now I want to stop here and let's go back. Sorry. This is like an overview of mitosis, right? Where we go from 1 cell into 2 cells. Now this cell is exactly the same DNA-wise that these 2 cells are. This cell is identical to the one on the left. So mitosis happens when you are growing, when you get a cut and you need to heal, when you're just maintaining your life and your cells need to replace themselves. That's mitosis. Now when we get to meiosis then, what we're going to do is we're going to start with one type of cell and end up with something different. The cells become different from each other. We start with 1 diploid cell and we get 4 different haploid cells. Now, we break meiosis into these 2 kind of stages, maybe, and each with 4 phases. Now, the 4 phases are the same that we learned: pro, meta, ana, telophase. But in meiosis 1, in the first set of those 4 phases, the homologs separate, and in the meiosis 2, the sister chromatids separate. So we'll kind of go through that right now. So meiosis, all right, we're going to start in interphase again. This is pretty much the same as mitosis, right? But we're thinking about cells now. Remember, meiosis is only happening in the sex organs. In humans, this is only happening in the testes or the ovaries. Now, chromosomes and the cell contents, again, double, like you would imagine. And here in prophase 1, we have a real big difference. So we're going to think of these chromosomes. This is, we'll say, chromosome number 1. It's the big chromosome. We get 1 from mom and 1 from dad. Now we'll be sexist and say the blue is from dad and the red is from mom, right? And what they do essentially is, in this phase, the chromosome number 1 that you got from dad actually kind of overlaps the one from mom, and right here what you see is it like crosses over and it switches. It'd be like basically grabbing your neighbor's hand and switching hands, okay? What that ends up with then is you start with 1 chromosome from dad, 1 chromosome from mom. They kind of lay across each other, and now you have a chromosome that is part mom, part dad, part mom. Here is part dad, part mom, part dad. They're different now, right? These chromosomes might be a little different. So if this is chromosome 1 and there's a gene for eye color on here, maybe your mom has green eyes and your dad has brown eyes. Or whatever it is, right? Now, we're starting to like shuffle the deck of DNA. And this is creating variation already. The chromosomes, just like before, line up in the middle of the cell. But the difference from mitosis is that in metaphase 1 of meiosis, the microtubules are attached to homologs, not sister chromatids. So this is chromosome that is mostly from dad, chromosome number 1 that is mostly from dad. This is chromosome number 1 that is mostly from mom, right? And what you see is when they pull apart here in anaphase, now you've got chromosome 1 over here and chromosome 1 over here, where the homologous chromosomes have separated already. And what you can see is after telophase, you've got a haploid cell already. But the DNA itself is still doubled. So essentially the same thing then happens again in meiosis 2, just like mitosis. Meiosis 2 is just like mitosis. The thing is, you've got different chromosomes here from when they separate. Then you can see that this one was fully mom, this one is part mom, and a little bit dad there. And you get then 4 different cells. So we can see that shown here as, one long red and then a short red with a little bit of blue. And we don't, in all the rest of the cells, we don't see a long red anywhere, right? So in meiosis, what you end up with is 4 different cells. And this is where you get differences in siblings, right? So ingredients come from the same mom or the same dad, but this essentially means that every single sperm a man creates is different. Every single egg a female creates is different. Whereas in mitosis, you start with one cell and you end up with a cell that is identical. And these cells are, this cell is identical to this cell, and this cell is identical to where it came from, basically. So there's 2 places where variation comes from, right? And the first thing is independent assortment. Okay, this is the idea of basically lining up. Where do the chromosomes line up? Now in this possibility number 1, we've got all the chromosomes from dad lining up on the left and all the chromosomes of mom lining up on the right. But there's another possibility. It's essentially completely random whether you have, the dad on the left or the dad on the right. And in this case, what we've got is dad on the left, mom on the right, mom on the left, dad on the right. So, even without crossing over, you still get some variation. I think even with 46 chromosomes, 23 pairs of chromosomes, you can have 64 million different combinations, right? And then, let alone crossing over then. When crossing over is another way we get variation, where the homologs no longer are wholly, paternal from dad or maternal from mom. So they're these 2 reasons are why sperm are, different from, each sperm is essentially unique. Each egg is unique. All right, with that, I hope you have a better understanding. If you have further questions, feel free to contact me. Another good way would be essentially to look up on YouTube, and you'll find plenty of videos that kind of have a summary of mitosis and meiosis. All right, with that, I'll see you later.